Hello, good morning. Myself, I am BSR. Today, our topic morphology. Plant morphology within 2 minutes. All of you stay with me for 2 minutes. I will give the complete data about the morphology. Okay. First, you take the root structure. So, you take this the root point of view at the tip, root cap, just above the root cap, meristematic tissue. Above the root meristematic tissue, there is elongation zone, then maturation zone. So, first, the root is covered by root cap. So that root cap is called a calyptra. At the tip, meristematic zone is present which contain meristematic cells which can divide continuously. So after that meristematic zone, elongation zone is present. So in the elongation zone, the cells undergo elongation helpful for vertical growth of the stem. Later on maturation zone, there only root hairs and lateral roots are forms. The root hairs are unicellular exogenous, whereas the lateral roots are endogenous multicellular. Lateral roots are developed from the pericycle. Then you go for root modification. Here you just look at the in the NCRT. According to NCRT, we are discussing. Please concentrate only on NCRT examples. So when you like it, you please subscribe immediately. I will give all the topics, all the subtopics within 2 minutes. Please remember. Okay, please subscribe also. Storage roots example, carrot, turnip. Just especially in the carrot and turnip, taproot stores food materials becomes tuberous. Whereas in sweet potato, adventitious root stores food materials. Then you go for mechanical roots are there in the stilt root and prop root. Stilt root example, sugarcane, maize. Especially in the sugarcane and maize, roots are developed from the lower nodes. Prop roots example, banyan tree, there from lower branches, adventitious roots are forming which grow towards the soil and gives mechanical strength. Nematophores example, Rhizophora abyssinia. In nematophore, Rhizophora, there the adventitious roots are growing negatively geotropic. The tip of the nematophores containing pores, they are called nematodes, which helpful for exchange of gases. When you go for stem modifications, the first one you look at their underground stem modifications. Example rhizome examples look at their ginger and turmeric. Especially these stems grows horizontally below the soil. Then you go for colocasia and jamnakan. There the stem grows vertically below the soil. Stem tuber example potato and bulb example allium. And so aerial stem modifications. Wiry coiled sensitive organs are called tendrils. Example, cucumber, pumpkin, watermelon, grapevine. And look at their thorns. These are the protective structures, hard woody pointed structures, mainly protective structures. Example, citrus and bougainvillea. Then phylloclades, generally in xerophytes, the phylloclades are present. There, the leaves fall off, stem is modified into green expanded structure called phylloclades. Flattened phylloclades seen in Opuntia, cylindrical phylloclades are present in Euphorbia. Subaerial stem modifications, example, runners look at their grass and strawberry, whereas stolons, example, jasminum and mint. Suckers, example, you look at their banana, pineapple, chrysanthemum. Especially in the stolons, there the branches are growing obliquely downwards. In the suckers, the branches are growing obliquely upwards. And then look at their offsets, example, Fistia and Icornia. So then when you go for leaf point of view, sheathing leaf bases are present in monocots and pulvinous leaf base. The leaf base is spongy in legume. And pinnately compound leaves are present in neem. It means only one rachis is present. On either side of the rachis, leaflets are present. That is called a pinnately compound leaf, example neem. Whereas a palmately compound leaf, example silk cotton. Silk cotton, then the petiole bears many leaflets. That is called palmately compound leaf. Then you go for phyllotaxy. Alternate phyllotaxy is present in China rose. Only one leaf develops at every node. Whereas opposite phyllotaxy, example Calotropis and Guava, their two leaves develops at every node. Rolled phyllotaxy, example Nerium and also Alstonia. There many leaves are developed at every node. When you go for leaf modification, tendrils seen in pea, spines seen in cacti, storage leaves seen in garlic, phyllodes seen in acacia, and especially in the insectivorous plants like peaches and Venus, flytrap, leaf modifications are present. When you go for inflorescence, here flower is a modified shoot. What is the difference between racemose inflorescence and cymose inflorescence? In the racemose, the peduncle grows indefinitely, and flowers are arranged in acropetal manner. Whereas cymos, the peduncle grows definitely, flowers are arranged in basipetal manner. So that is the difference. Whereas when you take in lily plant, there is no differentiation like calyx and corolla, that is called perianth seen in lily. When the flower cut into any plane to get equal halls, that is called actinomorphic. Example, chili, mustard, datura. Here, Malvaceae, Brassicaceae and Solanaceae. Zygomorphic condition means the flower can cut into only one plane to get equal halls. Example, Gulmohar and beets. It means Gulmohar, Cisalpinaceae, beans, Fabaceae family. Then you go for asymmetric flower. That flower cannot be cut into any plane. That is called asymmetric flower. Example, Kenna. Then you go for hypogynous flower, perigynous flower and epigynous flower. Hypogynous flower, their ovary is superior. Example, China rose, mustard, brinjal. Perigynous flower, there the ovary is half inferior, half superior. Example, plum, rose, peach. Epigynous flower, ovary is inferior. Example, guava, cucumber. Then you go for parts of the flowers. There especially we have to discuss about valvate estivation. When the perianth lobes are arranged with the small gaps, that is called valvate estivation. Example, calotropis. When the perianth lobes overlap one above the other, that is called a twisted estivation. Example, China rose, lady's finger, cotton, Malvaceae family members. Then you go for imbricate. Example, Acacia and Gulmohar. There, especially 1 plus 1 plus 2 arrangement, 1 plus 1 plus 3 arrangement. One is completely inside, one is completely outside. Three are overlapping. That is called imbricate. Example, Acacia and Gulmohar. Vexillary, Papilionaceous corolla. There, Papilionaceous corolla, standard petal. Below the standard petal, two wing petals are present. Below the wing petal, two keel petals are present. Example, Fabaceae. Then you go for androecium point of view. Staminodes, example, Acacia, and look at their epipetalous condition seen in brinjal, Solanaceae family members. Epiphyllous means here the epiphyllous means the stamens are attached to the tepals is called epiphyllous. Example, lily. Monodalphous, if stamens fuse to form one bundle called monodalphous. Example, China rose. Whereas diadalphous condition, two bundles. Example, P. Many bundles can be seen in citrus, that is called polyadalphous. Whereas filaments, it means the stamens are various in various length. Example, salvia and mustard. When you go for gynoecium, there the carpels. When the carpels are free, called apocarpus. Example, lotus and rose. When the carpels are fused, called syncarpus. Example, mustard and tomato. And you go for placentation point of view. Marginal placentation seen in P. Parietal placentation, mustard and argemone. Axial placentation seen in China rose and tomato, lemon. And whereas when you go to free central placentation, example, dianthus, prime rose. Basal placentation, ovule developed from the base of the ovary, is called basal placentation. Example, sunflower, marigold. And drupe fruit. Here, drupe fruit is a true fruit. Their drupe, especially outer epicarp, is leathery, middle mesocarp fleshy or fibrous, inner endocarp is stony. That's example, mango and coconut. Especially in the mango, edible part is a mesocarp, whereas in coconut, endosperm is the edible part. When you go to for seed point of view, in the ovule, outer layer, testa, that is developed into outer layer of the seed. Here, inner layer, that is called tegmen. Scarab, the ovule developed into hilum. Then micropyle is the apical part of the ovule that is developed into seed pore. Non-endospermic dicot seeds, example, Fabaceae. Generally monocots are endospermic, but non-endospermic monocots, example, orchids. Alluron layer can be seen between endosperm and embryo, and what is called scutellum, that is the shield-shaped cotyledon. Especially plumule in the monocot covered by coleoptile and radical covered by coleorhiza. Thank you to all. You please remember all these examples before examination, just before grand test. You just observe it. Okay, thank you to all. Please subscribe my channel. Thank you.